All right. We left off, at least as far as I could remember, we left off with a discussion of the structure of thick and thin filaments in the sarcomeres. That we have a sarcomere. The sarcomere goes from one Z line to the next Z line. Looking at this through a light microscope, what we would see is a dark band running down the center of the sarcomere. That is where the thick filaments are stacked on top of each other, alternating with the light bands, which are where we find stacks of thin filaments. The I band is the light band, the A band is the dark band. It was. Because I'm pretty sure we covered that. We talked about that. Yeah, well, we haven't gone into a lot of detail here yet, but now we are. All right. So, and I told you that muscles contract to produce force by shortening. And they can do that because the fascicles get shorter, because the muscle cells get shorter, because the myofibrils get shorter, because the sarcomeres get shorter. When a muscle is contracting, when a muscle cell is contracting, the Z lines are going to be brought closer together. The length of the sarcomere is being shortened up. But that is not occurring because the thick and thin filaments are getting shorter. Rather, there is an interaction between the thick filaments and the thin filaments that is going to cause the thin filaments and thick filaments to slide past each other. So if we think of my elbows as the Z lines, right? So when the muscle cell is at rest, they're this far apart. When we stimulate the muscle cell to contract, the thick filaments and thin filaments slide past each other. The ends of the sarcomere are being brought closer together. The sarcomere is shortening, but the thick and thin filaments are not getting shorter. The thick and thin filaments are sliding past each other. And this is called the sliding filament theory of muscle contraction. And that's about where I could remember that we left off. Okay, so now we got to tell you how that happens. So here we have a very close-up picture of thick and thin filaments. Thick filaments, shown here, are made of many, many, many individual molecules of a protein called myosin. Now, myosin molecules have kind of an interesting shape. I usually think of these as shaped kind of like hockey clubs or golf clubs, hockey sticks or golf clubs. What we see is each myosin molecule has this long, straight, like handle to it, which are all kind of neatly bundled together on the thick filament. And then right at the end, the protein makes this kind of change in angle, and part of the molecule sticks up out into space. This section is what's called the myosin head. It looks vaguely disturbing, but there you go. All right, so dozens, if not hundreds, of individual myosin molecules all bundled together with their little myosin heads sticking up out of the thick filament. The thin filaments have a little bit more complicated arrangement of molecules Most of the thin filaments are made of these rounded molecules. These are also proteins. Much of this are made out of individual actin protein molecules. Now, actin is a globular protein. It tends to fold up into a rounded shape. So in this illustration, the actins are shown as these kind of ping pong ball sorts of dealies. Any one thin filament is going to have hundreds, if not thousands, of individual actin molecules. And they've all been kind of pushed together to form this long chain of individually round ping pong ball kinds of structures, kind of like cramming a bunch of pearls together on a necklace. In addition, the thin filaments have two more proteins. This one shown here is this kind of long, stringy, dark purple one, is a protein called Tropomyosin. Tropomyosin is a fibrous protein, and you can see in this picture that it's kind of wrapped around the actins. So when you push the actins together, they form this kind of helix, this kind of depression or groove. And the tropomyosin is just kind of strung along these grooves, wrapped over and around the actins. The third interesting protein here in the thin filaments, I had to color one of these in because it's very hard to see in the rest of the illustration. These are molecules of something called troponin. The troponin molecules, at least in the way they're usually illustrated, look a little bit like footballs or rugby balls. Their one end is attached to the tropomyosin. So one end of the oval or one end of the, there's a word for this shape and it just went right out of my head.
No.
Yeah, see, it's this pill shape.
Elliptical?
Sort of, yeah. It's ellipsoid, maybe, is the word I'm looking for. I'll think of it about 3 o'clock this morning. One end is attached to the tropomyosin, and the other is just kind of projecting up into space. Right? So it's kind of at this cocked angle here with one end attached to the tropomyosin, and then the tropomyosin is wrapped around the actin, and altogether those form a thin filament. All right. So in order for the muscle cell to contract, we're going to have some type of interaction between the thick filament and the thin filament. So they will slide past each other. So the way this works is that on the myosin heads of the thick filament, there's an enzyme that breaks down ATP. So a molecule of ATP will bind to the myosin head, and the enzyme will break that down, but it's going to hang on to the pieces. Typically, to release the energy of ATP, we snap off the third phosphate. But in this case, it's going to break down the ATP to form a myosin ADP phosphate complex. Now, a real quick way of symbolizing a phosphate is a P with a circle around it. So the myosin head will bind a molecule of ATP, break the ATP, but will hang on to the phosphate and the ADP, forming a myosin ADP phosphate complex. Now this complex has a very high affinity for actin. So the myosin head, once it has the phosphate and the ADP, that myosin head is going to attach to a molecule of actin at a specific binding site. On the actin. So there's a spot on the actin molecule where the myosin is going to want to attach. And when the myosin head has an ADP and phosphate with it, it will do that. It will attach to the actin. Okay? This is going to now make a connection between the thick filament and the thin filament. And this is what's going to be called a cross bridge. Now, once the myosin head is attached to the actin, it's kind of like a juggling act. It's got, the myosin head's got a phosphate, the myosin head's got an ADP, and now it's trying to attach to the actin, and something's got to go. So the phosphate is released. That's going to allow the myosin protein to use the energy of the ATP molecule. The phosphate is released, and the myosin head is going to very abruptly shift its position. Now, the books aren't showing this quite the same way I learned it, and I'm going to stick with what I know, even if it's not exactly right. So here's a thick filament, and the myosin head has lifted up, and it's attached to a thin filament. The way I learned this was that the attachment was at a 90-degree angle. Maybe not, but that's the way I learned it. Now, when the phosphate is released, the myosin head is going to very abruptly shift from a 90-degree angle to a 45-degree angle. So, that's true. Myosin head has lifted up, and it's attached to the actin at a 90-degree angle. When it lets go of the phosphate, it shifts to a 45-degree angle. And you see what happens to the thin filament. It gets pulled across the thick filament. This is something called the power stroke. And the thin filament is going to slide across the thick filament, moving closer to the middle of the sarcomere. Now the ADP is released, but the myosin is still attached to the actin. To get the myosin head to let go, we need another molecule of ATP. That will bind to the myosin head. That will break the cross bridge. So here we are. A new molecule of ATP has to come along and bump off that myosin-actin connection. So the myosin head will pick up a new molecule of ATP. It will let go of the actin. It'll drop back down toward the thick filament. It'll recock to a 90-degree angle. And the myosin head will break down the ATP to form a myosin ADP phosphate complex. That complex can then lift back up and attach to a new spot, a new actin on the thin filament, forming a new cross bridge. It lets go of the phosphate and power strokes from a 90-degree angle to a 45-degree angle, moving the thin filament even closer toward the center. So it lifts up, attaches, power strokes. New ATP comes along, it lets go, recocks, reaches up, forms a new cross bridge, power strokes again, and so every time it power strokes, it's moving that thin filament a little bit more toward the center of the sarcomere. As long as the muscle cell is being stimulated, as long as the thick filament myosin heads can find a way to attach to the actins, it will continue to do this, cycle after cycle, stroke after stroke. And every thick filament has dozens of these myosin heads projecting out at different angles, so they're all capable of attaching to one of the thin filaments that's in the area. Are you kind of with me?
Mm-hmm.
Okay. So as long as the muscle cell is stimulated to contract, thick filaments will attach to thin filaments and form cross bridges, power stroke and slide the thin filaments. That's going to shorten the sarcomere. That's going to shorten the myofibrils. That's going to shorten the muscle cells. Okay, that's great, but what are these other two guys doing here? Okay. Your muscles are not constantly in a state of contraction. Your muscles are relaxed, and then when you require them to contract, because these are skeletal muscles and under voluntary control, they are stimulated to contract. So there has to be something regulating when the cell will contract and when it won't. Something has to be saying, yes, you can, no, you can't. The regulatory protein, the one in this group that is controlling whether or not cross bridges can form, whether or not contraction will occur, is actually the molecule troponin. Troponin regulates whether or not cross bridges can form. Now the way it does that is through this attachment that it has to the tropomyosin. When your muscle cell is relaxed, when it is not actively contracting and cross-bridging and power stroking, this tropomyosin molecule is actually lying on top of the actin's binding site. I don't know if you can see it, but in the picture, there's a little red dot on each of these ping pong balls. And that is where the myosin head wants to attach. If the tropomyosin is in the way, the myosin cannot form a cross bridge with the actin. So when your muscle cells are relaxed, the tropomyosin is kind of lying in this groove, physically blocking the ability of the myosin to attach to the actin. When the muscle cell is stimulated to contract, when it's going to contract, what we're going to see is a very rapid increase in the concentration of calcium ions in the sarcoplasm. So when the muscle cell is stimulated to contract, there's going to be a very rapid increase in the amount of calcium floating around inside the cell's cytoplasm. calcium ions bind to the troponin. Where they attach, so calcium ions floating around, floating around, floating around. The calcium ions are going to attach themselves right here at the free end of the troponin. So by adding on an atom of calcium at the end of the troponin, that makes this side of the molecule a little bit heavier, right? So it's kind of at an angle like this, and then we put on a calcium, and now it starts to go like this. It starts to kind of tip over or flop over. When it does that, this side of the molecule is attached to the tropomyosin. So as the calcium ions in the cell increase and bind to the troponin flops over and drags the tropomyosin out of the groove. And that's going to expose the binding site on the actin. Now, the myosin head with the myosin ADP phosphate complex can attach to the actin, forming a cross bridge at a 90-degree angle, release the phosphate so it can power stroke to a 45-degree angle, and slide the thin filament across the thick filament closer to the center of the sarcomere. As long as the muscle cell is being stimulated to contract, calcium concentration in the sarcoplasm stays high, calcium stays bonded to the troponin stays flopped over and keeps the tropomyosin out of the way. When the stimulus stops, when the muscle is going to be allowed to relax, the calcium concentration goes back down to the normal range. So calcium ion concentration starts to go down. The calcium comes off of the troponin. The troponin flips back up into its original position and pushes the tropomyosin back into the groove, blocking the actin and blocking the binding site. And so now the muscle cell can relax. No more cross bridges, no more power strokes, no more force or tension. Calcium goes up, cross bridges can form. Calcium goes down, cross bridges can't form. We don't have the whole story yet, do we? Because where is all that calcium coming from? And then where does it go?
Doesn't that just tie into what you taught us before?
Not exactly. There's a little bit more to the story, right? You see, I told you earlier that each of these myofibrils is completely surrounded and separated from the other myofibrils by this elaborate internal network of membrane, an organelle that in muscle cells is called the sarcoplasmic reticulum. It's analogous to the endoplasmic reticulum. And it turns out that in skeletal muscle cells, the sarcoplasmic reticulum let's see if I can get it. Acts as a reservoir. I love the French. Reservoir, if you're not French, right? Reservoir for calcium. That organelle is going to suck up calcium as it's available and store it inside the sarcoplasmic reticulum. When the muscle cell is stimulated to contract, that stimulus causes calcium gates in the sarcoplasmic reticulum to open. There's a high concentration of calcium inside the organelle and a lower concentration of calcium outside the organelle. So calcium ions diffuse out and bind to the troponin. The troponin flops over, dragging the tropomyosin out of the groove and exposing the binding site on the actin of the thin filaments. The myosin heads of the thick filaments can reach out, attach to the actin, forming a cross bridge, moving from a 90-degree angle to a 45-degree angle, power stroking and sliding the filaments. When the stimulus stops, calcium pumps in the sarcoplasmic reticulum begin to suck the calcium back out of the cytoplasm of the cell, pulling it back inside the organelle. Calcium comes off of the troponin flips back up, pushes the tropomyosin back into the groove, and blocks the binding site of the thin filaments from being able to form cross bridges with the thick filaments. And the stimulus and the contraction stops, and the muscle cell relaxes. But we're not done yet. Our skeletal muscle fibers have very large and elaborate sarcoplasmic reticulum organelles. Each part of it surrounds and covers and separates all of the myofibrils. Now, these muscle cells also have another feature we don't find in many other cells. And you can see them here. The darker orange is part of the sarcoplasmic reticulum. The lighter beige here is actually connected to the cell membrane. It's part of the sarcolemma. And you might have noticed in this picture that there's these little holes here and there along the cell membrane. These are actually openings into these structures. These structures are called transverse tubules, or T-tubules. These are extensions of the sarcolemma. They're like little tunnels, little subways, little underpasses that will go from one side of the cell all the way across to the other side. So this little hole is an opening into the T-tubule. So we have the sarcolemma, and we have continuous with it, part of it, the T-tubule. Give you a bigger picture here. All right. So here's the sarcoplasmic reticulum, and here's the T-tubule. Now it turns out that the T-tubules, although the picture doesn't show it that well, the T-tubules don't actually touch the sarcoplasmic reticulum. They're very, 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 very close together, but they're not actually in physical contact with each other. And yet, excitation, stimulation of the transverse tubule can be transferred in some mysterious way to the sarcoplasmic reticulum. I keep using the fairly vague word stimulus here. What actually sets all of this off is action potentials. There they are, all right? Skeletal muscle fibers can experience action potentials, these waves of electrochemical phenomena. Stimulated to contract, an action potential will roll down the cell membrane of a muscle cell. But it's also going to dive down into the T-tubules. And as that action potential goes through the T-tubules, it's passing very, 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 very close to the sarcoplasmic reticulum. And somehow that irritates the sarcoplasmic reticulum. It's like that game you used to play with your brothers and sisters, right? I'm not touching you. I'm not touching her. Mom, I'm not touching her. I don't know what she's complaining about. I'm not touching you. Isn't that irritating? Oh, yeah. So the action potential going through the T-tubule irritates the sarcoplasmic reticulum. The sarcoplasmic reticulum opens up its calcium gates. Calcium ions flood out into the sarcoplasm, bind to the troponin. The troponin flops over, drags the tropomyosin out of the groove, exposing the actin. So the myosin head can form cross bridges, power stroke from a 90-degree angle to a 45-degree angle, and slide the thin filaments closer to the center across the thick filaments. As long as action potentials are going down the cell membrane and through these T-tubules, the sarcoplasmic reticulum keeps its calcium gates open, and calcium levels inside the cell's cytoplasm stay high. When the action potentials stop, no more action potentials are coming down this cell, then the sarcoplasmic reticulum will close its calcium gates, start up its calcium pumps, pull the calcium out of the cytoplasm. The calcium comes off of the troponin flips back up into its original position, pushing the tropomyosin back into the groove and blocking the binding site of the actin, and no more cross bridges can be formed, and no more sliding of the thick and thin filaments. Action potentials. How about that? You didn't believe me when I said we get back to it, it would actually start to make some sense, but there it is, action potentials. Now, this gets even better. Oh, how could it possibly get better than this? You see, there's a funny thing about skeletal muscle, and we did mention this back when we were talking about tissues. Skeletal muscle is not normally spontaneously contractile. Skeletal muscle is also called voluntary muscle because you have conscious control over its contraction. You send a signal from your brain, from the
Control center?
post-central gyrus? No, precentral gyrus. That's the motor one, right? Precentral gyrus is the motor one. We'll know in 2 weeks, right? Okay. So your cerebral cortex sends a message down through your spinal nerves, and it says, okay, guys, it's time to contract. So your brain sends a signal down your spinal cord, and the spinal cord will send these action potentials out through your neurons that are part of your nerves, and those neurons are going to now communicate with these skeletal muscle fibers. The fibers of skeletal muscle don't normally, under normal physiological conditions, just contract on their own. They have to be told to contract by your nervous system. Now, that's not to say that sometimes things don't work the way they should. Everyone's had twitches, cramps, spasms. Those would be abnormal physiological responses. But ordinarily, to get your muscles to contract, they have to be told to do it by a neuron. Now, I have a couple different pictures here. So if we get in really close, if we get into high magnification, here is the end of the neuron's axon. And this is the skeletal muscle cell. Now they communicate with each other, again, without being in physical contact. They don't actually touch. So where we get to the end of the axon, what we see is this kind of enlarged bulb at the end of the axon. Now that goes by a couple of different names. In some books it's called the axon terminal. And in other books it's called the synaptic knob. The Saladin book calls it a synaptic knob. I'm not sure what the OpenStax book is calling it. This is the illustration from the Saladin book. So what we see is that there is this enlarged area at the very tippy end of the neuron's axon, the synaptic knob. This is the skeletal muscle fiber, and there's actually a very small gap between the two. That little gap is called the synaptic cleft. The gap. On the other side of the gap, on the other side of the synaptic cleft, we have the cell membrane, the sarcolemma of the muscle cell. Now this particular area of the membrane has some special features. So this section of the muscle cell membrane is called the motor end plate. Together, the synaptic knob, the cleft, and the motor end plate form a structure called the neuromuscular junction. Now, muscle cells can have action potentials and neuron cells can have action potentials. They're both excitable cells. So I want you to imagine that an action potential has been started up in the pink neuron. Right? Action potentials move down the cell, so that action potential moves down the neuron's axon, all through, down the nerve, until it gets to the axon terminal, the synaptic knob. So the action potential is coming down this way, and it gets to the synaptic knob. As the action potential is passing through the membrane of the knob, that just sounds rude, doesn't it? It's got a knob, right? As it moves through the membrane of the synaptic knob, so we have the A action potential in the membrane of the synaptic knob. That's going to open calcium gates in the neuron's cell membrane. Now, extracellular fluid has a slightly higher calcium concentration than cytoplasm. So calcium from the extracellular fluid diffuses in the knob. Now, one of the reasons why the knob is kind of larger and lumpier than the rest of the axon is located down here inside the knob are thousands and thousands of little tiny organelles, little membrane-bound packets that are called synaptic vesicles. Now these organelles, these little bubbles of membrane inside the synaptic knob, contain many, many molecules of a substance called acetylcholine. Now, acetylcholine is where we're going to be using a lot, so I can show you that there's a fairly typical abbreviation for acetylcholine, capital A, capital C, lowercase h. So you don't have to keep writing out the whole word over and over and over again. The synaptic vesicles contain many, many molecules of acetylcholine. When the action potential comes through the membrane of the synaptic knob and calcium gates in the knob membrane open up, calcium comes into the synaptic knob, and that triggers the movement of some of these synaptic vesicles. Not all of them, not all at once, but a few of these guys are going to get moved to the inside surface of the cell membrane. And then exocytosis is going to occur. So that bubble of membrane is going to get popped open like a bad zip. And out is going to come the acetylcholine. So as calcium diffuses into the knob, that triggers the movement of the vesicles, or at least some of them, and exocytosis, secretion of acetylcholine into the synaptic cleft. Now the next part of this, unfortunately, I'm going to have to draw because I don't really have a big enough picture to do this justice. Now, one of the things that makes the membrane of the muscle cell on this side of the synaptic cleft kind of special is it has many, many membrane proteins, thousands and thousands of membrane proteins that are acetylcholine receptors. That's in the sarcolemma of the motor end plate. We don't see very many of them in the rest of the membrane, but right here at the motor end plate, at the neuromuscular junction, there's hundreds of them. Now, I told you guys that when an action potential gets to the end of a cell, it's going to just get to the end and stop, right? So you got to keep in mind that the action potential that's in the neuron has gotten this far and it can't go any further, it's done. But as it has come through, it has triggered the release of some of these acetylcholine molecules into the synaptic cleft. And on the other side of that cleft are hundreds of acetylcholine receptors. So the acetylcholine molecules will diffuse across the cleft and they will bind to the acetylcholine receptors. Now here's the kind of the cool part, although you guys probably don't think so. Usually at the motor end plate, we find very close by the acetylcholine receptors other membrane proteins that are sodium gates. when a molecule of acetylcholine binds to an acetylcholine receptor, the receptor is going to elbow the sodium gate next door, and the sodium gate is going to open briefly. And a little bit of sodium is going to then enter the muscle cell. Sodium ions are positively charged. So when the sodium gate opens, even if it's just very briefly, a few sodium ions are going to come into the cell, bringing with it their positive charges. That depolarizes the muscle cell just a little bit. Now, these are actually such small changes that what we're talking about here are graded potentials, not big changes, graded potentials. In this location, these graded potentials are called end plate potentials. So a molecule of acetylcholine binds to an acetylcholine receptor. The receptor nudges the sodium gate next door. The sodium gate opens briefly. A few sodium ions enter the muscle cell, and the muscle cell is slightly depolarized. It experiences an end plate potential. But graded potentials show summation. They have temporal summation, and they have spatial summation. So a molecule of acetylcholine gets onto an acetylcholine receptor, and we get an end plate potential. And another molecule of acetylcholine gets onto another acetylcholine receptor, and we get another end plate potential. And another molecule of acetylcholine binds to another acetylcholine receptor, and we get another end plate potential. And maybe the first one down here is over, and the acetylcholine has been cleared away, and so this one is going to hit again and again and again and again. Temporal summation, spatial summation, end plate potential, end plate potential, end plate potential. It builds up and it builds up and it builds up until we get to threshold. Woohoo! And the muscle cell is going to have action potential. And the action potential sweeps down the sarcolemma and dives down through the T tubules and irritates the sarcoplasmic reticulum. And the sarcoplasmic reticulum throws open its calcium gates, and calcium ions flood out into the sarcoplasm, binding to the troponin, and the troponin flops over and drags the tropomyosin out of the groove, exposing the actin so the myosin head can reach up and form a myosin ADP phosphate complex cross bridge with the actin. It releases the phosphate, power strokes from a 90-degree angle to a 45-degree angle, sliding the thin filaments across the thick filaments. And then another action potential comes down the neuron, and it releases more acetylcholine, and the muscle cell has another action potential, and it keeps happening and happening and happening, and waves and waves and streams and chains of action potential go through that muscle cell, and the power, it keeps forming cross bridges, and it keeps power stroking, and it contracts. When the action potentials stop, I'm not done yet. I don't know where you people are going. When the action potentials in the neurons stop, it's not going to release any more acetylcholine. No more acetylcholine is binding onto the receptors, and there are enzymes that kind of break this down and clear these guys away. There is a cleanup system here, which means that the muscle cell doesn't get depolarized anymore, which means it doesn't have any more action potentials. So the sarcoplasmic reticulum starts pumping the calcium back in. The calcium comes off of the troponin flips back up, pushes the tropomyosin back into the groove, blocking the actin. No more cross bridges, no more power strokes, no more sliding of the filaments. Wow, what a story. All right. Now, this whole story I told you is called excitation-contraction coupling. So, you don't have to go through this. You don't have to name it all these different things. The whole story. Action potential in the neuron, calcium comes into the synaptic knob, synaptic vesicles move to the membrane, exocytosis of the acetylcholine, acetylcholine binding to the receptors, depolarization of the muscle cell membrane at the motor end plate, action potentials in the muscle cell, sliding filaments, that whole thing goes by the name of excitation contraction coupling. It's exciting.
No pun intended.
Yeah. Just happy accident. Okay, now I have a little bit more I want to tell you about this system. You'll notice here when I mentioned the red neuron that as it's getting closer to the muscle, this neuron's axon actually splits into several different tendrils, and each one of these is communicating with a different muscle cell. So each branch off of the axon has its own synaptic knob, its own neuromuscular junction with a different muscle cell. Action potentials coming down the red neuron are going to stimulate this cell, this cell, this cell, and this cell. All four of these are going to be stimulated to contract. This grouping is something called a motor unit. A motor unit is all of the muscle cells that are controlled or stimulated by one neuron. Now, this picture that they're giving us is actually fairly simple. So and of course you guys know that the neurons are not red and blue, right?
Yes.
Okay. All right, just want to make sure. All right, so these four red, pink muscle cells are all being controlled by the red neuron. They are one motor unit. But we also have here a blue neuron that does the same thing. Its axon forms branches, and it's going to form a neuromuscular junction with this cell and this cell and this cell and this cell. So the blue neuron is controlling the blue muscle cells. This is a different motor unit. Action potentials coming down the red one are going to cause the red cells to contract. Action potentials coming down the blue one are going to cause the blue cells to contract. If we have action potentials coming down both of them, all 8 cells are going to contract. Now this, like I said, this is very simplified. Typically, real motor units may have anywhere from a few hundred to a few thousand muscle cells in them. So one neuron might be triggering, stimulating 100, 200, 500, 1,000, 2,000, 3,000 individual muscle cells. This explains a phenomenon we're already all very familiar with, which is that you can control how much force you get when you contract a muscle, right? So, biceps brachii, right? Mm-hmm. Biceps brachii. Right, so if I activate a few motor units, I get a very slight low force contraction. If I activate more motor units, I get a stronger, more forceful contraction. And if I activate even more motor units, I get a very strong and very forceful contraction. All right. So this phenomenon of being able to adjust how much force you produce out of any one muscle is called recruitment. Typically when you're faced with some sort of task, so you have to do something with your muscles, mentally you kind of imagine, evaluate, use your prior experience to estimate how many motor units it's going to take, how much muscle force it's going to take to do that task. So let's say your mother-in-law is coming for a visit, right? So you have to drive down to Orlando International and pick her up at the airport, And so you drive down there and you come around the arrivals gate, and there she is out on the sidewalk with her suitcases, waiting for you to come and pick her up. So you pull over to the curb and you pop open your trunk and you get out and you're like, oh, Mother Jones, I'm so glad to see you. We're so happy you could come for a visit. Let me help you with your suitcases. And you're thinking, she's only come for a week. How heavy can these guys be? So you mentally make an estimate of how many motor units you're going to need in your muscles to lift up her suitcases. And you reach down and you grab those handles and nothing happens. Mother Jones, did you pack gold bricks in here? What have you got in here? Well, mentally you are already recalculating. It's like a GPS, recalculating. So you're going to try this again. You reach down, you take another grab, and that time the suitcases come up easily. You have recruited more motor units into doing this job. As far as I know, it works, the word applies if you go back the other way. So if you think you got to move or pick up something really heavy, and it turns out to be unexpectedly lightweight, damping down, turning off motor units is also called recruitment. You've had that experience, I'm sure.
Like somebody throws a box at you, pretending that it's heavy.
Yeah, you're like, oh, and then of course it was actually full of air or pillows or bubble wrap or something. Yeah, or yeah, so you picked up something expecting it to be a full mug, and actually you drank it all already, and so, but you adjust. Now that would also be called recruitment. All right, I'll quit there. You guys were